How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Keira Deneen. I'm a certified genetic counselor and your host. Last episode, we were joined by Dr. Eric Green, the director of the National Human Genome Research Institute at the NIH, and we chatted about the recent completion of the human genome. So we're continuing the conversation this week. So I'm joined by the co-founders of the Telomere Consortium, which led to this project. Not helped, you guys led the project. So I'm joined by Dr. Karen Miga, who is an assistant professor in Biomolecular Engineering at the University of California, Santa Cruz. She's also the associate director of the UCSC Genomics Institute. And Dr. Adam Phillippy is a senior investigator and head of the Genome Informatics Section at the National Human Genome Research Institute. Thank you both so much for joining me on the show to continue the conversation about this feat in genetics.
Thanks so much for having us. Yeah.
So to catch listeners up, especially if they didn't hear our last episode with Dr. Eric Green, the first draft of the human genome was completed in 2003. So there were major headlines then, which is a little bit confusing for some people with the recent announcement that we just completed the human genome sequence. So can one of you enlighten us, like, what is the difference? What did we actually complete this time for those that are maybe skipped our last episode?
You want to lead it off, Karen?
Sure. The big announcement in 2003 that we've celebrated was really the parts of the genome that we knew were gene-rich, or the euchromatic portions that were celebrated to be 99% complete. It was recognized at the time that about 8 to 10% of our genome that we knew were in these regions that were known to be heterochromatic and repeat-rich and gene-poor were excluded from that announcement. So the scientific community largely knew back in 2003, but the celebration continued. And I think that as you mentioned, there is this sense of confusion because of that. What we're talking about today are these large persistent gaps that have been spanning regions of our genome that we know are incredibly important for the function of the cell. These include regions such as the centromeres that we know are important for chromosome segregation as cells divide, large regions that span things called segmental duplications or gene families that are expanding and contracting, entire short arms of all 5 acrocentric chromosomes. This is 13, 14, 15, 21, and 22. And these really encapsulate as well the ribosomal rDNAs, which have been off of our ability to do research for decades now on these very important regions. So I think the reason these parts of our genome were not included before was not to recognize that they weren't important. I think everyone understood their importance. It was just technology wasn't there at the time to put those pieces together and make an accurate map, and we're able to do that today.
Yeah, and we kind of knew roughly what was missing and roughly where it was located in the genome. It was just really the level of resolution was at the microscope. We were looking down the microscope, seeing the heterochromatin and the euchromatin and how they stain differently and how they were packaged differently. But it's really drilling down to the level of the ACGs and Ts that was missing. So if you think from like the map analogy, we had like Google Earth looking at the whole countries, and the whole globe. And our project was drilling all the way down to get the street level map, every last ACGT accounted for.
That's a great analogy to think about it. Like, okay, we can see all the continents, like all the chromosomes, right? They're all there.
Yeah, exactly.
All right, but like how deep can we get in and how confident are we about the sequence of those genes? I think that's a lot of what you're talking about. But it sounds like it was, 8 to 10% of the genome that was not previously sequenced, that's what you guys finished in your teams?
Yeah, at the end of the completion, the finishing stage of the Human Genome Project, it was about 8 to 10% that was left unfinished. And that was represented as a placeholder in the references. Since then, if you downloaded HT19 or GRCH38, you would get millions and millions of Ns as just unknown characters in these regions that Karen mentioned, like the centromeres and the rDNAs and so And so when I first saw that as kind of a bioinformatician and admitted perfectionist, my FASTA file has all of these ends in it. Like, why is this not finished? Like, what's in here? How can we finish the job?
And I'm sure that passion and that eagerness of just like, let's get it done is what, drove you to be able to finish this. PerkinElmer Genomics is a global leader in genetic testing, focusing on rare diseases, inherited disorders, newborn screening, and hereditary cancer. Testing services support the full continuum of care from preconception and prenatal to neonatal, pediatric, and adult. Testing options include sequencing for targeted genes, multiple genes, the whole exome or genome, and copy number variations. Using a simple saliva or blood sample, PerkinElmer Genomics answers complex genetic questions that can proactively inform patient care and end the diagnostic odyssey for families. Learn more at PerkinElmerGenomics.com. Are you developing a new therapeutic? Need a partner to help? No matter where you are in your development program, Worldwide Clinical Trials is available to guide you. With more than 35 years of global experience founded on medical science, Worldwide will execute your trial with confidence. Worldwide Clinical Trials has an extraordinary depth of medical expertise in key therapeutic areas, including cardiovascular, metabolic, neuroscience, oncology, and rare diseases. Unique logistical challenges and lack of precedents can make rare disease trials intimidating. Worldwide offers customized insight, deep expertise, and novel solutions to get the most out of your efforts. Worldwide uses their extensive network of global resources to meet or exceed patient recruitment and retention goals. This is achieved by combining the benefits of global scale with personal service and senior management accessibility. Stay tuned for our full episode interview with Worldwide here on DNA Today. Learn more about how Worldwide can be your clinical trial partner at worldwide.com. Want to become a genetic counselor? Looking for ways to engage with the field and boost your resume for grad school applications? Then you should check out Sarah Lawrence's Why Genetic Counseling Wednesday Summer Series. Every Wednesday this June, Sarah Lawrence is hosting this series where you can interact through Zoom with genetic counselors from different specialties. It kicks off on June 1st. You can sign up at slc.edu/DNA today. Again, visit slc.edu/DNA today to register to level up your resume for applications in the fall. Was there ever a thought, like maybe back in 2003, was there ever a thought we're not ever going to complete the genome? Like it's almost a pipe dream at that point? Or were we like, at some point we're going to be able to finish it? Like, I know you guys probably weren't involved at that point in the field because you're talking 20 years ago, but, from talking to colleagues that were involved at that point, what were their thoughts?
Yeah, if you read some of the original papers, you kind of get that feeling that they thought was almost impossible. And it was kind of written off like, well, we'll never get this. And we all kind of agree we'll never get this. And since some of the most rewarding conversations I've had with some of the old timers that were involved with the original project is, wow, like we're so glad that you've uncovered this, because I just was always told through my career, don't even bother with these regions because it's so difficult and complex that you'll just be wasting your career trying to figure it out. And so having them make those comments has made this all the more rewarding.
And just to comment there as well, I think both Adam and I started our career kind of with the first announcement of the initial Human Genome Project. And I came from a very special lab, which is really crediting another co-lead of our working group, Evan Eichler, who was one of the initial researchers involved in the Human Genome Project that made it extremely clear that this project was not done, that there was important sequence content that was missing from the map. And really to credit him, for decades, he had taken that soapbox and established a lot of the cell lines and tools that the Telomere Consortium actually used to reach the finish line. And also working with pioneers in the satellite field. These are the tandem repeats that were missing. I think there are a lot of folks, not in the major spotlight, but on the sidelines that have been working for decades for this moment, building more and more maps to help us understand, as Adam mentioned, that beautiful metaphor, of what we would expect when we do hit the ground level. And I think that was with this anticipated reach that we would meet this milestone. It was just a matter of time. It was just a matter of technology.
It's kind of a cool segue for how Karen and I first met and started working together, that like I was completely naive. I saw these ends and I was like, let's finish it. Like, I'm coming from a technical perspective. And Karen was like, one of the few people that knows what actually we expect to see in some of these regions. And so, if we take the explorer analogy, like, she's my trusted guide saying like, here, okay, here's what we're going to expect to find in here. And then, seeing what we actually expected to see was also very gratifying and gave us confidence that we were on the right track.
Especially in research, and it can be, so disheartening sometimes to not be able to be making progress. So, I mean, this is just monumental. Why was it so difficult to sequence this last 8 to 10%? I imagine back in 2003, obviously we didn't have the technology we do today. What's some of the technology that led to the ability for us to do this? And if there's other aspects too that, allowed the human genome to actually be completed?
Yeah, it's 20 years of continual technology development, both on the sequencing side and on the algorithmic side, developing the methods that can actually stitch these genomes back together again. And I always give the analogy of a puzzle when we're trying to do what we call genome assembly and reconstructing one of these genomes. And if your puzzle has bigger pieces, you have fewer pieces and they're easier to put together again. And that is a perfect analogy for what happened here, that we had emerging long-read sequencing technologies from PacBio and Oxford Nanopore, and they're somewhat complementary in their strengths at the moment, and we were able to develop new methods that could leverage the strengths of both of those technologies and effectively put together the puzzle in these most repetitive, most difficult regions of the genome.
And so these are like the ultra long reads that you're referencing, like just to have those bigger puzzle pieces, as you're saying, as opposed to the really short reads that we were using more back in the day. Am I getting that right?
Yeah, exactly. To put firm numbers on it, Human Genome Project was Sanger-based technology, 100 to 700 base read lengths, pretty good accuracy on each of those reads. But, the most common kind of repeat size in the human genome is like 5 kb, 7 kb, 10 kb. And so the reads just aren't long enough. You can't tell always which copy of the repeat it's coming from when it's just a few hundred bases. And then we went through.
So you couldn't line it up kind of thing because there's just
exactly.
so many repeats. Yeah. I didn't understand that till now.
So if you kind of just take a read and imagine, can I figure out where this goes in the genome? If you can't figure that out, you can't assemble the genome.
Right. If the puzzle piece is identical in so many different ones, then how are you going to fit it in? You don't know where it goes.
That's exactly right. And when you have the Nanopore ultra long reads that are up to 1 million bases long,
whoa,
they have a high probability of capturing some of those unique variants that lets you uniquely place it in the genome.
So we went from, like you said, 180, something like that, to now 1 million?
Yep.
Wow.
Exactly.
But I'm sure what Adam was also going to emphasize is that it's not only these extremely long, which is where we led in with the consortium. Adam and I started teaming up to generate a large high coverage of these very extremely long or ultra long sequences. But while we were doing so, there was new technology that came out from another company known as PacBio. This generated high fidelity reads from a circular consensus model. And this model, you're not reading a giant sequence in terms of hundreds of thousands to millions of bases. You're reading the same sequence over and over again. And at the end of the day, it just turns out to be incredibly accurate. So in that model of thinking about this as a puzzle, if there are slight flecks of color that can help you distinguish where that piece should go, that you wouldn't be able to see if it was kind of a more granular, gaze at the puzzle pieces. This allowed our team to really hone in on small details that we couldn't see before with the longer reads. And I think that both of these elements, the 20,000 base pair, highly accurate, near perfect read, and these very, very long reads were really the answer that technology provided.
And it turned out that 10,000, 15,000 is kind of the magic threshold of a read length to where it becomes possible to assemble the human genome. And so when PacBio came out with Hi-Fi and they could push 20 kb perfect accurate reads, the reason we couldn't assemble with perfect accuracy when we had Illumina is that we topped out at 200, 300 bases. And it was really when we got to tens of kb that it was possible. So we took those 20 kb perfect reads and combined them with the megabase pair 95% accuracy reads, and together they got the job done.
And obviously it's in the title, telomere to telomere. What is so unique about the telomeres? What can we learn about now that we have this sequence? Because, as we said, as Adam was saying, it's like a bunch of ends. We don't know what is the base here. I think about, in terms of my line of work, being a prenatal genetic counselor, I think of chromosome 21, of having people that have 3 copies of chromosome 21 of Down syndrome. And this is one of the acrocentric chromosomes that Karen mentioned in terms of having really tiny arms on the top of the chromosome. So a lot of this sounds like was not sequenced before. We already know a lot about Down syndrome. It's one of the most well-known genetic conditions. But are we going to learn even more information about Down syndrome because of these newly completed sequences?
Well, one interesting finding. Oh, go ahead, Adam.
You go ahead first, Ken.
Well, Adam, if I could give him credit, has spent a tremendous amount of time studying the acrocentrics, and I have spent a tremendous amount of time studying the centromeric satellites, and there's probably stories for you in both of those aspects, so maybe we could both answer here. From the centromeric satellite team, we were really interested in chromosome 21 because we were now making estimates of all of these tandem repeats where the kinetochore sits. This is where the centromere actually functions.
Like the belts of the chromosome.
Exactly yes. Right. There's a series of proteins that sit there, and that's really the critical mark where these proteins sit on the genome. And we are starting to characterize all this tandem repeats known as satellite DNAs that exist there. And if you look at all the other chromosomes, what we typically find is that long array of tandem repeats sometimes can extend 3 to 5 million bases, megabases. I'm sorry. But if you're in chromosome 21, for example, this was our smallest centromere. It was like 10 times smaller than all the others. And so that instantly started to, raise some interest from our group, just thinking, how does the size, the genomic organization of these sites, really contribute to our understanding of chromosome stability and centromere function? And I think your question about trisomy 21 is perfectly placed for these types of new maps and new information about centromere structure and how that could play into better understanding the role in something like Down syndrome or trisomy 21.
Yeah, and I was just going to point to the beauty of these acrocentric chromosomes, and that I found one of the most rewarding parts of this project is to see this kind of quilt work pattern of interleaved segmental duplications and satellites, which was a bit of a surprise to me. I'm sure, cytogeneticists and satellite experts, kind of knew this going in. But it's an alternating pattern of kind of gene-rich seg dupes interleaved with these satellite repeats. And all 5 of the short arms of the 5 acrocentrics are so incredibly similar to one another that if you to just take a random 5 kilobase window of sequence from any of the acrocentrics, you will find that exact same sequence on average at 99% identity in one of the other acrocentrics. And so they're all incredibly self-similar to one another. And chromosome 21, as an example, is also frequently associated with Robertsonian translocations, which if you have, can predispose you to have a child with Down syndrome. And those Robertsonians are most commonly between chromosomes 13, 14, and 21. And we found a highly conserved, segmentally duplicated kind of core structure within those short arms that's incredibly similar between those 3 chromosomes. And so there's some relationship there between that high level of identity between the sequence and the frequency, I think, of those fusion events. And so now that we have the precise sequence identity, like Karen mentioned for the centromeres, that extends all the way through the short arm of the chromosomes, we can use that sequence now to plan experiments, to kind of build human artificial chromosomes, to do all sorts of new experiments to better understand how these events take place and their consequences.
It's really a jumping board of like, okay, we figured out the sequence, but like now think about everything that we can do and learn from this. And that's interesting. I'm surprised I didn't think of Robertsonian translocations like prepping for this interview, because I have my undergrad in cytogenetics, and obviously Robertsonian translocations come up a lot in prenatal as well, and that makes sense that they have the same sequence on those small arms of the acrocentric, and why that they would be combined for some people, because those are probably just lining up a lot because they're matching at that point. Yeah. So that's a really astute point there. If you're applying to genetic counseling grad schools in the near future, I highly recommend checking out Sarah Lawrence's Why Genetic Counseling Wednesday Summer Series. This is the 3rd year Sarah Lawrence will be hosting this series, where you can interact through Zoom with genetic counselors from different specialties for 2 hours every Wednesday in June. It kicks off on Wednesday, June 1st at noon Eastern. As many of you know, I graduated from Sarah Lawrence's program 2 years ago, and the series was a fun way to interact with prospective genetic counseling students. I'm really looking forward to meeting more of you during this series. Myself and fellow genetic counselors will share about our roles and answer your questions live. Not only will you hear from genetic counselors in a variety of specialties, you'll also have the opportunity to discuss ethical and social implications of genomic medicine, engage with current students, and learn about the exciting present and projected future of the profession. Anyone who attends all 5 will earn a certificate of completion and receive an application fee waiver for the grad program. Register now before we are fully booked. Go to slc.edu/DNA today. Again, that's slc.edu/DNA today to be part of this interactive genetic counseling experience. Are you seeking a career with purpose in the healthcare space? I'd recommend checking out Worldwide Clinical Trials, a contract research organization that partners with pharmaceutical and biotechnology companies to advance development of new medicines. Apply your passion at Worldwide, where they take an entrepreneurial approach backed by scientific expertise to help bring treatment options to patients. With a global team of 3,000 experts, you'll be inspired by bright thinkers, dreamers, and doers who innovate and invent for a brighter tomorrow. Perks include unlimited flexible paid time off, limited travel requirements, and the ability to work remotely. Bring your talents and passion and join the team at worldwide.com. Did you know PerkinElmer Genomics was one of the first laboratories to offer whole genome sequencing on a clinical basis? Whole genome sequencing can maximize clinical diagnostic yield for patients. With turnaround time of 4 weeks for the proband sample, PerkinElmer's whole genome sequencing test is designed to provide access to additional valuable information compared to an exome. PerkinElmer also offers prenatal whole genome sequencing as well as ultra-rapid whole genome sequencing for critically ill newborns using dried blood spots. The ultra-rapid genome has a turnaround time of 5 days and includes mito, chromosomal CNV analysis, STR-TNR screening, and biochemical analysis. Also, listen back to episode 176 with Dr. Madhuri Hegde, where we explored the power of whole genome sequencing, which also happens to be one of my favorite episodes of DNA Today. And stay tuned for a couple more episodes with PerkinElmer soon. Discover all that PerkinElmer Genomics has to offer at PerkinElmerGenomics.com. There's basically 2 types of DNA material. There's the heterochromatin, which is the non-coding parts, and then there's the euchromatin, the coding genes that we've mentioned so far. So for these repetitive sequences, does it contain mostly one of these types? I think you guys were hinting at this, but I just wanted to dive in a little deeper.
Well, I would just make the clarification up front that heterochromatin can have coding material in it. And as Karen has schooled me, heterochromatin is epigenetically defined.
Genetics,
so
it's a lot complicated than we thought, right?
Yeah, and so the thing that surprised me is the amount of genes that we found, say, on the short arms, where most of the new genes that we found live. And yes, most of them look like they're silenced and not expressing, but they are genes and highly similar paralogs to known genes elsewhere in the genome. And in rare cases or in disorders like cancers and so on, they might be activated and expressed. And so that's something I'm very interested in following up on, is measuring the expression and the epigenetics of these paralogs that typically live silenced in heterochromatin and what happens to them if things go wrong or even during different developmental stages.
And we'll have to update the textbooks then on the definition there, since I am outdated. My education's already outdated.
Yeah, I'm sure, as you know, there's multiple kinds of heterochromatin. When we think about our cells going through different cell types and differentiating, and the most persistent gaps were the parts that were typically associated with constitutive heterochromatin, or the parts that were typically packaged densely throughout multiple different cell types. And I think that that's really interesting because, as your question alluded to, there is, it doesn't matter if you're looking at human genomes or non-human genomes, or if you're looking at fish or plants, there's a trend to where the sequences that are organized or enriched in these regions are largely composed of tandem repeats or satellite DNAs, a lot of tandem repeats and satellite DNAs. And these can extend for 10 to 20 millions of bases. They're typically, have embedded transposable elements and segmental duplications that contain genes. They're just a jungle of repetitive DNA. And so that is a trend, a genomic feature. I think it's highly specialized. It's something that we haven't really studied in our textbooks very well. We've been thinking about, genes and promoters and things are kind of in this, intergenic, what is this DNA doing? And there are large parts of our genome that are just beautifully repetitive, that we have just not been able to study before. And they do trend with this type of epigenetic regulation. And the fact that there are these regions of our genome, as Adam mentioned, that that look like normal textbook genic organizations that are composed of the seg dupes that are plopped into this, unique and spectacular part of our genome, that demands a new chapter in a textbook to really understand how that could be selected for or against, or how that plays into cellular function and evolution.
Yes, we're rewriting the textbooks, I think, as we talk with everything. And that just kind of makes me also think about, with all this repetitive sequences, have we even had time yet, because this is so new, but have we had time to compare this new reference genome that we have to other species to see, okay, this is highly conserved, meaning that we see this in a lot of other species and that it must be very far back in the evolutionary tree. Or is this something that maybe we'll do in the future because you guys just finished this weeks ago? So,
well, there was a beautiful paper by Glynis Logsdon focused on chromosome 8, which provided a nice first example of thinking about how to study centromeres, through the lens of comparative genomics, looking across great apes. Evan Eichler as well had been working for some time at looking at non-human primate comparisons of these large segmental duplications. And Mitchell Volger featured that work as well in his paper. So there are a couple of our companion papers that did go into some of these non-human comparative projects. I think Adam can probably speak to a little bit more of a future gaze here too, because I don't want to speak for him, but it's had an incredible role outside of human, the Vertebrate Genome Project and various other projects looking outside of humans to try to elevate all of the similes to be more complete.
Yeah, the bit I'm most excited about is understanding this as kind of a new mutational class, that when we think about looking at mutations in the genome, people have been studying, SNPs and small indels for the last 20 years. But there's this really cool, curious association that the segmentally duplicated sequence always is kind of co-associated with these satellite repeats that Karen's interested in. They kind of move around the genome hand in hand. And I almost think of it as kind of like, potentially innovation of evolution to copy and paste a big bit of the code of the genome and then modify it, and maybe it becomes useful in a future iteration. So maybe you think if you're writing a document, I'm going to make a new abstract for a new meeting. I'll copy an old abstract to a new document, modify it a little bit, and then have something new. This, I think, type of copy, paste, and rearrangement in the genome is a whole kind of new mutational class that we can now study with these T to T genomes. And so, like Karen mentioned, the Eichler Lab, very interested in the role of segmental duplications in the evolution of the primate lineages. And so we're kicking off a project now with Evan and Katerina Makova at Penn State to do T to T genomes for kind of all of the high great apes that we're familiar with, like chimp, gorilla, bonobo, and so forth. And so I think that'll have some really fascinating findings when it comes to the evolution of these species.
yeah.
We definitely have expectations of how the centromere should be positioned, because these, you can see, as you mentioned, your expertise in cytogenetics, we've been able to really track these events. And they move around. There's centromere repositioning, and the satellites classes we've been able to see by fish. So much like the story for human, there's a lot going into these non-human primate maps to get to that base level of building off of expectations once again.
Yes. And Adam, you brought up an interesting point with just classification, and where my brain was also headed was how variant classification might be impacted by now having this finished genome. How do we see this changing in the future? Well, the terms like variant of uncertain significance, is that someday we're going to be looking back as we're going to be like old people with walkers and everything when we say, oh, remember when we used to say VUS? Like, is that going to be outdated at some point because now we do have reference genomes? Or is that just like total dream? Are we always going to have variants of uncertain significance?
Yeah, I mean, and I don't mean to insinuate that, we're finding completely new forms of variation. It's variation that we knew about, we just couldn't measure it very well. And so like copy number variants in genes, rearrangements, inversions, and so on, we're just able to really nail them down accurately and precisely now. And so the hope is that we will be able to associate some of those more large scale and copy number variants to diseases and phenotypes in a way that we've been able to do so far, primarily with SNPs and indels. And so it's unlocking those additional classes of variation. But things get really complicated really fast when you get into the acrocentrics and other copy number variable regions, to the point that you might have to kind of think at an allele level, like this genome has these 20 versions of this particular gene or this allele, and, they're roughly in these locations. I've seen this before in a previous case, and it had this particular effect. And that really requires us to now sequence as many human genomes as we can with these methods so that we can start to associate all of these novel structural variations with particular diseases or other effects.
Yeah, I think that's a good point, especially that we need to be looking at, we're not done here. Like it's a major accomplishment, but we also need to keep sequencing more and more genomes because everybody's a little bit different. So certainly points that variants are different. Go ahead, Karen.
Oh, I was just going to say, what we're celebrating is the ability to really read the genome and more completely and comprehensively. But reading the genome and understanding the genome are two different things. And I think for the past 2 decades, we've been able to look closely base by base and start to motivate functional analysis and associations, building off of that map. And this is the first time we're, as you mentioned, being able to really dive into these regions. And even if we handed the genomics community 1,000 of these complete genomes, there's still a tremendous amount of work that needs to come out of the research community to make sense of it, to start actually studying what it means to be a variant of, associated with a function versus unknown significance. And there, I think, are a lot of association and new technologies that need to develop on that front too, to really connect the dots.
Yeah, and I think one question that people listening might have is, what about patients that have done a whole genome sequence where they were told, all right, we're looking at your entire genome, we've sequenced it, this is what we found, and have had those conversations, especially in the pediatric realm is usually where we're seeing this. How much of their genome is actually sequenced with this clinical whole genome sequence? And in the future, should that be re-looked at? Will we be sequencing even more of the genome, similar to what you guys did?
Yeah, well, if we're talking about kind of a short read, Illumina-based whole genome sequencing, you are sequencing the whole genome. Illumina is spitting out sequences for really everything that's there. It's just your ability to map that to the reference and to identify that there's a difference here or a variant compared to the major allele or some reference genome. And so we have a companion paper that looks at the improvements that are to be gained by mapping those short read data sets to this complete reference genome. And even older data sets that were generated with short read Illumina can identify thousands of additional variants per genome by mapping to a complete reference. And so there is some additional gain from the old data, but still, when we find variants there, at present day, we won't be able to really say what the significance of those variants is. And so this is, to Karen's point, that now we can explore this additional 8%. We need to redo a lot of association studies and redo a lot of experiments to understand the significance of the variants that we're seeing. And then once that gets added to the database, at some point in the future, we'll be able to call variants and associate with diseases and give predictions in those locations. But that's going to be a gradual process over the next decade to figure out what these variants are doing in the genome. But yeah, then I think it'll fold back into standard clinical practice, and we'll just have a more comprehensive and accurate prediction based on your genome.
And it sounds like for some people, they may be able to go back to that raw data, but then analyze it again with the reference genomes we have, or maybe they don't actually have to run the sample again. Maybe in the future they will. But from what you're saying, it sounds like our first step might just be reanalyzing and comparing to the reference genome. Definitely
for a lot of these big cohorts that were collected and sequenced with the idea of doing association studies, because half the battle, 90% of the battle there is just getting the samples and getting the sequencing done. At an individual patient level, I think it'll make more sense 5-10 years from now to just redo your genome with the new technologies and get the accurate picture. It's really kind of the data sets that were expensive to collect in the first place. Yes, we can go back and reanalyze those and get some added gain. But for everything from here on, we should be using the new long read text to get the complete picture.
Well, anything else to add to that? I have learned so much in this interview, I have to say. It's just been a pleasure being able to hear directly from both of you on how this works and what you were able to do and where we're headed with it, too. I think we covered so much. And for those that didn't listen to tune back to the episode before this one with Dr. Eric Green, because we go over some of the more basic level. I think today we really, I got to dive into more of like, what are all my questions about this? So thank you both so much. I really appreciate it. And congratulations on this amazing, massive accomplishment.
Thank you. And thanks for having us.
Yeah, thanks, Kira. I would close by saying that we really benefited from 30, 40 years of research investment in sequencing technology and computational technology. I was trained as a computer scientist, and so I kind of see how much these two fields rely on one another. And we couldn't have accomplished this without advances in both the computational side and on the wet lab side. And we're just, standing on the shoulders of giants here, benefiting from all of that prior work. And, we're in the right place at the right time with the right expertise and the right collaborators to get the job done.
For more information about today's episode, visit dnapodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@dnapodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Kira Deneen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me are all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.